further back to the next specimen, and here we, again, go with what you know, go with your landmarks, right? So major landmarks are going to be notochord, neuroectoderm, right? This is going to be the skin ectoderm here. Okay, coming off of the neuroectoderm right at the top, that's why they call it the crest, are the neural crest cells. When the neural tube forms, it forms a crest of cells called the neural crest. And these ectodermal cells migrate through the body. And they're going to give rise to structures like the ganglia, the peripheral nerves, the adrenal medulla, for example. Okay? Dorsal, ventral, right? This is now our heart. So now we've reached the heart. This is the ventricle. And between the ventricle and what is going to become the spine and the neural tube is the foregut. This is going to become the esophagus. And this here is going to be dorsal aorta. Okay? As the body forms, remember this is a chick. So it is forming from a blastodisc where there's been a primitive streak, and the cells have, from the epiblast, have moved through that permanent streak and formed these three germ layers, right? So this is flattened somewhat. And the edges are going to start curling in. And those become the lateral body folds. This one right here, as it comes in, they become the lateral wall, the lateral walls of the body. Okay? Go to the next section, right? In the next section, we've got dorsal aorta here. We've got the foregut here. We start to make note of the vitelline veins out here. Vitelline veins are going to be associated with what structure?
Yolk sac.
Crickets.
Yolk sac.
Yolk sac, yes, thank you. From back in the cheap seats. Yeah. The yolk sac, right? And then what's this over here? This is the beginning of one of the spaces. We refer to those spaces as coeloms. It's going to be important to understand which are the extra-embryonic and the intra-embryonic coeloms. The reason being is the extra-embryonic coelom will get bound by those structures that we will refer to collectively as the fetal membranes or the extra-embryonic membranes, like the choroid and the amnion, okay? And the intra-embryonic coelom is going to give rise to the pleural cavity, peritoneal cavity, and the pericardial cavity. I'm going to see that. Hopefully we'll learn about that tomorrow as we move into the next topic in lecture. Now we're going to move further back. And again, go with what you know. Notochord right here, right? Here's our neural canal. Here's our neural tube, right? Right here. This is our neural tube. Here is our dorsal aorta. And we start to get a coelomic space right here. Here's one of those spaces starting to form, a coelomic space. What are these out to the side?
Vitelline veins.
Vitelline vessels, exactly. And here we start to have another space. This space, however, is bound by tissue. This becomes the midgut. This becomes the midgut. So we're beyond the heart, and we're into the midgut. We're into the peritoneal cavity. This space, when these coelomic spaces all fuse, they're going to give rise to the intracoelomic space. And at this level, it would differentiate into the peritoneal cavity at this space. But right now, they're coelomic spaces. And this is a somite. And we're going to talk about that because the somites form from aggregations of cells along a tube of mesodermal tissue that lies on either side of the neural tube. It's called the paraxial mesoderm. And the paraxial mesoderm gets its name because it lies alongside the axis, the craniocaudal axis, which we now know is defined by has been defined already by the primitive streak, and it's going to be defined by the notochord extending cranial to caudally, as well as the neural tube extending cranial to caudally along that primitive street. So these are the somites, and the somites give rise to the body segments of the body.